Hello, everyone, and welcome to today's webinar titled Rapid Discovery of Therapeutic Antibodies for SARS-CoV-2 and Other Viral Pandemics. This webinar is a part of the ongoing coronavirus virtual webinar series. I am Michelle Ashton of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and sponsored by Berkeley Lights. For more information about our sponsor, please visit www.berkeleylights.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Anupam Singhal, the Product Manager of Antibody Therapeutics at Berkeley Lights. For a complete biography on Dr. Singhal, please visit the Biography tab at the top of your screen. Dr. Singhal, you may now begin your presentation.
Humans are susceptible to a myriad of different pathogenic viruses, including HIV, influenza, Zika virus, Ebola, and the severe acute respiratory syndrome coronavirus 2, known as SARS-CoV-2. These viruses can result in a wide range of diseases with symptoms ranging from common flu-like symptoms to death. Development of therapeutics against each virus has its unique set of challenges, including rapid transmission, high mutation rates, and delayed onset of symptoms due to long latency times. The current COVID-19 pandemic is a dramatic example of the challenge of curtailing the rapidly transmitting SARS-CoV-2 virus. In a matter of months, COVID-19 turned from a regional disease in Hubei province, China, to a global pandemic that has infected greater than 500,000 people and killing over 20,000 as of March 26, 2020. These numbers are continuing to expand daily. In response to infection, the human immune system produces antibodies that bind viruses and target them for clearance. Unfortunately, some exposed individuals do not produce viral neutralizing antibodies, which may result in serious life-threatening diseases. In these instances, administration of neutralizing antibodies, known as passive immunotherapy, may protect individuals from infection or enable them to recover post-infection. As a result, the rapid discovery of antibodies against viruses is a promising strategy for combating viruses. For the SARS-CoV-2 virus, antibodies targeting the spike protein may prevent binding of the virus to the human ACE2 receptor, thus preventing infection. Opto-B-cell antibody discovery on the Beacon system bypasses inefficient hybridoma fusion by directly screening B cells from immunized animals or human patients. B cells are harvested and purified from diverse B cell compartments, including peripheral blood, spleen, bone marrow, and lymph nodes. These cells are functionally screened on the Beacon system, and antigen-specific sequences are recovered in less than 1 week. The Beacon system fully automates cell sample handling, screening, cDNA synthesis, and sequence recovery. A 96-well plate containing a purified B cell sample is inserted into the Beacon. The Beacon system then automatically loads the cell sample into OptoSelect chips. Tens of thousands of cells can be screened in OptoSelect chips containing nanopens, each less than 1 nanoliter in volume, greater than 10,000 times smaller than a well in a 96-well plate. The small volumes enable secreted antibody concentrations to reach detectable levels in a matter of minutes. B cells are cloned into nanopens using Berkeley Lights' OptoSelect technology, in which cells are manipulated by light. This light-based manipulation can be performed in parallel across hundreds to thousands of cells. Several tens of thousands of B cells can be cloned across multiple chips in less than 2 hours. Nanopen chambers enable rapid, precise, and highly sensitive assays. In minutes, secreted antibodies from B cells can be assayed to determine antigen specificity. In this assay, the target antigen is conjugated to beads and mixed with a secondary antibody prior to importing into the chip. Secreted antibodies from B cells diffuse out of pens and bind the antigen-coated beads. The secondary antibody is then sequestered on the beads to provide a rapid fluorescent readout in a matter of minutes. This assay is performed on tens of thousands of cells simultaneously. In this field of view, hundreds of cells are screened to identify less than a dozen hits. The beacon enables down selection of lead candidates through multiple assays for antigen specificity and function that can be performed either in serial or in parallel. Better characterization up front reduces the expense of having to sequence or clone irrelevant non-functional hits. In a pandemic situation, there is often little control over the available patient samples. Opto B cell discovery enables screening of B cells from acute or convalescent patients. Plasma B cells can be enriched from fresh PBMCs obtained from acutely infected patients. Alternatively, memory B cells can be enriched from thawed frozen PBMC samples and activated off-chip in tissue culture prior to screening. We demonstrated the screening of frozen PBMC samples that were obtained from healthy human donors. The samples were thawed and memory B cells were purified by FACS or MAX. The memory B cells were activated in tissue culture for 4 days using a proprietary B cell activation media. The activated memory B cells were then screened on-chip to confirm antibody IgG secretion. We can identify antiviral antibodies from purified human B cells. Full-length viral proteins or their subunits can be conjugated to beads, and antigen-specific antibodies are detected using a fluorescently labeled anti-human secondary antibody. In the images here, we detected antibodies that were specific and cross-reactive against the full-length spike protein and receptor binding domain for SARS-CoV-2 virus. This work was performed in collaboration with Rob Carnahan and James Crow in their lab at the Vanderbilt University Medical Center. We have demonstrated a proof-of-concept viral neutralization assay using a vesicular stomatitis pseudovirus engineered to express the Ebola glycoprotein. We created a set of hex cell lines secreting either neutralizing or non-neutralizing antibodies and mixed these together. We then loaded single hex cells with vero cells into individual nanopens. We used our standard bead-based assay to confirm that the hex cells were secreting antibodies, and simultaneously, we identified pens in which antibodies blocked viral infection, as confirmed by the absence of expression of a fluorescent GFP marker in the vero cells. This assay demonstrates that we can successfully identify cells that are secreting viral neutralizing antibodies. After identification of hits, antigen-specific antibody sequences are recovered via unloading of cells or unloading of beads on which on-chip cDNA synthesis has been performed. After recovery, off-chip amplification of heavy and light chain genes is performed. In just 2 days, opto B-cell antibody discovery enables single-cell cloning, screening, on-chip genomics, and sequencing of antigen-specific antibody sequences. We have active collaborations throughout the world to develop new therapeutic antibodies and vaccines for COVID-19. Our collaborators have already leveraged the speed of our platform to discover greater than 500 antibodies to SARS-CoV-2 in a single day. Please reach out to us if you are interested to learn more and join us in our Global Emerging Pathogen Antibody Discovery Consortium in the fight against COVID-19. Thank you.
Thank you, Dr. Singhal, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Let's get started. Okay, your first question here is, let's see, can B-cell discovery on the Beacon system screen human patient samples?
Yes, so the opto B-cell discovery platform will work with both human B-cell samples as well as mouse B-cell samples. And it's compatible with fresh plasma B-cells as well as activated memory B-cells.
Great, thank you. How many antibodies can be discovered from a single B-cell sample?
We've demonstrated from immunized mice that we can discover greater than 5,000 hits per sample. And we leverage the ability to keep plasma B cells viable in tissue culture for multiple days so that we can perform multiple workflows on a single sample. And as mentioned in the presentation, we have collaborators who are working on COVID-19 who have discovered greater than 500 antigen-specific or viral-specific antibodies in a single day.
Great, thank you. Okay, it looks like your next question is, are all isotopes of immunoglobulins can be detected in B cells?
Yes, so there is, in principle, no restriction on which isotypes are assayed in this workflow. The selection of secondary detection reagent will determine which isotype is being assayed for.
Now, can you describe with memory B cells from blood samples, do they give you an idea about the mucosal immunity?
We haven't done any specific studies of mucosal immunity, but we do know that B cells can be screened from multiple different B cell compartments and organs.
Great, thank you. We've got lots of great questions coming in from the audience, and here's your next one. What is the efficiency of sequence recovery from B cells screened during the workflow?
Yeah, we've recently launched this OptoSeq BCR capability, which reliably recovers greater than 85% of paired heavy and light chain sequences from single B cells.
Great, thank you. Okay, your next one is, are you able to recover all cells from the chip, or do you notice that you lose cells during the process?
So we, by performing OptoSeq BCR, we now do on-chip cDNA synthesis on beads, and so we're now recovering beads rather than the cells. And our recovery process now is highly efficient so that we obtain sequences from greater than 80% of all antigen-specific antibodies of interest.
Now there's a second part to that question, and it says, how does the export of cells work technically?
Right, so as with the cell loading, we use this optoselect technology to bring beads or cells out into the channel and then activate the fluidics to export the samples into 96 well plates.
Now, do the antibodies change with the mutations of COVID-19?
So we've demonstrated that by performing multiple assays with different antigen variants, you can identify antibodies that are specific or cross-reactive against multiple targets. So in this case, if you were screening against different variants of the spike protein in a single experiment, a single workflow, you'd expect to find antibodies, again, that are specific to particular variants and cross-reactive against multiple variants.
Great, thank you. Okay, your next question here says, do you work with other surface proteins of SARS-CoV-2 for the antibody production?
Our own internal experiences have been with passing against the spike protein as well as the RBD of that spike protein for SARS-CoV-2. In principle, all of the other surface proteins of the virus can be assayed and antibodies can be selected for them. And I believe that we have customers that are targeting a wide array of different targets on the virus.
Now, following that question, can other surface proteins of SARS-CoV-2 be good target of antibody production?
We believe today the scientific community has identified that the viruses enter human cells using the viral spike protein, which is why this has attracted the greatest attention as a potential drug target.
Great, thank you. Will the antibody cause acute lung disease?
So there is still extensive preclinical and clinical testing required for any new drug therapeutic, including antibodies that are discovered on our platform or any other platform.
Are all surface proteins, do they induce antibodies?
So we're able to screen any sample from an immunized animal or human in which there has been an immune response.
Great, thank you. And I just want to remind the audience, if you'd like to submit a question, you can do so now. Just go ahead and type your question into the Ask a Question box located on the far left of your screen. As a reminder, if there's any questions we don't get to today, they will be followed up via email after the presentation is over. So we'll give the audience just a few minutes to submit some questions. If there are no further questions at this moment, I'd like to once again thank Dr. Singhal for his educational presentation. Do you have any final comments for our audience?
Yeah, I encourage you to seek us out if you're interested in our antibody discovery platform, and especially if you are interested in our current fight against COVID-19.
Great, thank you. And thanks again for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their questions. I'd also like to thank our sponsor, Berkeley Lights, for sponsoring today's webinar. Any questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand through the end of this year, 2020. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Please also visit the Berkeley Lights booth now in the exhibit hall to receive more information. Until next time, goodbye.